Hi everyone, my name is Samantha Quintanilla. I'm a senior here at the Butte Lab at UT Arlington, and I just want to talk about my project a little bit. So we know that mycobacteria elongate their cell walls by polar growth. We specifically are interested in learning more about how these proteins are regulated to control polar expansion. So this leads me to introduce my protein of interest, polar A. As we said earlier, mycobacteria elongate through the poles. Here we see that the old pole elongates more than the new pole. Then cells divide and the process occurs all over again. In figure 2, not much is known about polar A, but I found when I depleted this protein, we begin to see cell death after about 24 to 30 hours. When polar A is present in the cell, represented by blue here, cells are able to grow and survive, but when polar A is depleted here in red, cells are not able to survive. For this reason, we have concluded that polar A is essential in Mycobacterium smegmatis. In figure 3, when we looked at the cells that I had depleted of polar A under the microscope, we see short, fatter, bulgy cells down here. This points to the cell not being able to properly elongate. When we stain these depleted cells with HADA, which is a peptidoglycan cell wall stain, we see that delocalized staining. It's not at the poles like it is here at the septum, where we would usually see it. So because of this stain is a marker for peptidoglycan synthesis and remodeling. We have come to the conclusion that polar A is needed for proper peptidoglycan synthesis or remodeling at the poles. For figure 4, here we have polar A tagged with GFP, and although GFP is usually green, we have switched it to red in order to see localization better with the staining. So we see polar A localization is at the poles, and it is also a bit at the septum. When we quantified a signal in staining, we saw that localization is occurring at the poles, like we have mentioned before, but peptidoglycan synthesis is occurring subpolarly, so not at the poles, not at the tips. So what does this mean? We're not sure yet, but this could mean that peptidoglycan insertion is occurring subpolarly. But once again, we don't know yet. We still have many questions and experiments to follow up on this, but this is what we found so far. Thank you for listening.